Hello Studisians, welcome back to the channel Studies. So, in this video, we are performing an experiment in the Microbiology Lab in NIT ROURKELA, the aim of which is to prepare culture media using nutrient agar. So, let's start with the video. The material required will be Petri plate, flask, agar, LB broth, distilled water, and weight balance that you can see. This is the weight balance. And the procedure will be like 2.25 grams of agar was added and dissolved in the solution per 150 ml of distilled water. But here we had taken 300 ml of distilled water. So we have to double the amount of agar. So we took 4.5 grams of agar and 3.75 grams of luria bertini was taken and dissolved in 150 ml of distilled water generally. But here we took 300 ml of distilled water, and that's why we doubled the amount of luria bertini in the flask. The flask was shaken to mix the agar, and then the solution was first boiled in the oven for 2 to 2.5 minutes that I set, and yes, then wait for that. Now, carefully holding it, and let it cool for a few minutes, and then plug the flask's mouth with cotton, just like uh, making a tight cotton plug like this was my first time, so maybe it is not very good cotton plug. Now the flask is covered with the paper and secured it with a tie. A tight thread is tied across it. The flask was placed in an autoclave and sterilized it for 15 to 20 minutes at 15 psi and 121 degrees Celsius temperature, which is a good temperature to kill the microorganisms or bacteria, as you can say. And then, after waiting for 15 to 20 minutes, we went to the lab again and poured the sterilized solution into the Petri dish, filling up to 15 to 18 ml, half or three-fourth of the Petri dish. Generally, before continuing the experiment in the laminar airflow, we should first sanitize our hands completely and then set the Petri plates in an incubator and incubate them for 24 hours and then observe. So this was all about the today's experiment. So don't forget to like, share and subscribe the channel Studiesa. And if you have any query or any doubt, you can ask me in the comment box or you can DM me on Instagram. The link is given in the description box. So thank you. Bye bye. We'll see you in the next video.